Hello everyone, Assalamu alaikum. In this video lecture, I am going to discuss with you about the phase 1 reaction of drug metabolism. At the end of lecture, you may find the quiz for your self-assessment. For that, you have to attempt the answer in the comment section. These are the content of this lecture. At the end of this lecture, you will be able to learn about the different type of phase 1 reaction, the different type of phase 1 oxidation reaction, that is the carbon center heteroatom and their different type, and the reduction and hydrolysis reaction and the different type of drug that are being metabolized by these type of reaction. So bioorganic chemistry of drug metabolism. Chemistry of drug metabolism can be understood by the understanding the different type of reaction, that is the phase 1 and phase 2 reaction. So basically these two reaction overall contain the drug detoxification process, or sometimes they can be bioactivation process in case of prodrug. So that's why classified into the two different phases. Now let's discuss the phase 1 reaction. Phase 1 reaction are basically the functionalization reaction. The example of phase 1 reaction are oxidation, reduction and hydrolysis. The phase 1 reaction introduced the different type of polar function group in our drug molecule. Phase 2 reaction are known as the conjugation reaction. The examples of phase 2 reaction are glucuronide conjugation, sulfate conjugation and the amino acid conjugation. These reactions occur in the sequential manner. They are being catalyzed by the specific type of metabolic enzymes. Phase 1 reaction. Phase 1 reaction are known as non-synthetic reactions because they are basically involve the introduction of the different type of polar functional group in our drug molecule. The examples are oxidation, reduction, hydrolysis, cyclization, decyclization. These reactions are catalyzed by the specific type of cytochrome enzyme that are the mixed function oxidase enzyme. These enzymes mainly occur inside the liver. These enzymes tend to be the non-specific, catalyzing the metabolism of wide variety of the different compounds. The two common mixed function oxidase enzymes are the cytochrome P450 and the flavin monoxygenase, but mainly the cytochrome P450 are involved in the drug metabolism process. In these reactions, a polar functional group, that can be hydroxyl, that can be amino acid, amino group or any other, is basically introduced in our drug molecule, or it can be unmasked if already present. What is mean by unmasked means before metabolism the drug may have a specific type of polar functional group in their chemical structure, but it may be attached or in the certain site in the drug molecule, or it may be surrounded by certain type of non-polar functional group. So after metabolism, these non-polar functional group are basically removed and these polar functional group can become prominent. After the phase 1 reaction, the drug undergoes through the phase 2 reaction to get converted into more polar form, because most of the products of the phase 1 reaction are still not eliminated from the body. So that's why undergo phase 2 reaction to get converted into the more polar form, because for excretion the more polar form of the drug metabolite is required. Now let's discuss first reaction of the phase 1 reaction, that is the oxidation reaction. This is one of the most common phase 1 reaction. In this reaction, the hydrophilicity or the polar solubility of drug molecule is being enhanced by the introduction or by the substitution of polar functional group in their chemical structure, like hydroxyl functional group. This reaction can occur at the different site or places inside the drug molecule. So on that basis, oxidation reaction can be divided into different types, like it can occur at the carbon-carbon center, it can occur at the carbon-heteroatom center, and so on. Now let's discuss first the oxidation reaction that is occurring at the carbon center. The oxidation reaction of phase 1 that is occurring at the carbon center, it can occur either at the aromatic ring, means the aromatic or benzene ring that is present in the chemical structure of drug molecule. For example, the acetanilide get metabolized into paracetamol by this type of reaction. Olefinic center, that is the unsaturated double bond or triple bond that are present in the chemical structure of the drug. The example of drug that are being metabolized at this specific site are the carbamazepine. Aliphatic group, these are the alkyl side chain, and valproic acid is being metabolized at the site in their chemical structure. Other important type of drug that undergo the phase 1 oxidation reaction are the phenylbutazone, valproic acid, carbamazepine, minoxidil, and so on. Now let's discuss the aromatic hydroxylation. The aromatic hydroxylation is basically referred to the introduction of hydroxyl functional group in the aromatic ring of the drug molecule. So this reaction is also catalyzed by the mixed function oxidation enzyme and now the arene that are basically aromatic ring. Now after this aromatic hydroxylation, they are converted into the phenolic metabolite that are the arenol. Almost all the aromatic hydroxylation are believed to get converted into an intermediate state, that is the formation of epoxide ion. That epoxide ion is also known as arene oxide, which get rearranged and get converted into the arenol that are the phenolic metabolite product of this aromatic hydroxylation. So this is the figure for the epoxide intermediate ion. So what is happening basically in the metabolism of drug containing the aromatic ring? First of all, oxygen is being introduced at their aromatic or benzene ring. So this is how oxygen appears and it forms bond. So this ion is the epoxide ion. After this is the intermediate stage, and after that it will again rearrange itself and get converted into the phenolic compound. So now it will become OH. So that will be the phenolic metabolite is the arenol. Aromatic hydroxylation is basically major route for the metabolism of most of the drug molecule, because mainly the most of the drug molecule contain the phenyl group, that is the aromatic group. The example of such drug can be the propranolol, phenobarbital, phenytoin, phenylbutazone, ethinyl estradiol, warfarin, and many others. Most of the phenolic metabolite that are the product of the aromatic hydroxylation, clear, which are formed from the aromatic oxidation, undergo further conversion to the polar and water soluble form by the glucuronide conjugation or sulfate conjugation. Means over here I am referring to the first of all, when drug undergo through the aromatic oxidation, then it may undergo further towards the conjugation reaction. The type of conjugation reaction that is required for phenolic metabolite can be either glucuronide or sulfate conjugate, and after that the drug will become highly polar. The metabolite of drug become highly polar, and now they can excreted in the urine. For example, the major urinary metabolite of phenytoin found in the human is the O-glucuronide conjugate. The example is the para-hydroxyphenytoin. So this is the example of the diagram showing the metabolism of phenytoin. So this is the chemical structure of phenytoin. In this you may observe there are the two aromatic rings that are the benzene ring. So it is crystal clear that it will undergo through the aromatic hydroxylation. So now in the first step, that is the phase 1 oxidation, the phenytoin is get converted into para-hydroxyphenytoin. Para-hydroxy referring to the hydroxyl functional group is being introduced in their aromatic ring at para position. After this, this drug will undergo through the phase 2 conjugation reaction. The type of conjugation reaction will be the glucuronide conjugation because of this phenolic metabolite. So glucuronide conjugation is basically the involvement of the glucuronide molecule binding with this hydroxyl functional group. So in this diagram you may observe this is the O-glucuronide of the phenytoin has been produced, and now this will ultimately go through the process of excretion. Aromatic oxidation. Now there are the certain other factors that can influence the aromatic oxidation. Means it's a strength. Some may stimulate this process, some may reduce this reaction. Because there are certain type of functional group that are electron rich or electron rich activating group. Clear? So the ring activating group are basically facilitating the aromatic hydroxylation. Means those aromatic ring in which the electron rich functional group are present, in them aromatic oxidation reaction takes place at the higher rate. On the other hand, there are certain type of other functional group that are basically causing the deactivation of ring. They are mainly the electron withdrawing group. They are the electronegative group, like the chlorine, nitrogen, chlorine basically referring to the halogen family. Nitrogen are 3-carboxylic acid sulfonamide. These are basically the slow or resistant to hydroxylation. Means in those rings in which these type of deactivated functional group are present, they are causing the deactivation of ring. They are extracting the electron from the ring. Now the ring is becoming deactivated, so that's why they may causes the slower aromatic hydroxylation, or they may cause the resistance to the aromatic hydroxylation reaction. So this is the again example showing the aromatic oxidation. In this compound, this is the diazepam. You may observe the two aromatic rings are present. So in this case, the aromatic oxidation will take place at that ring where there is no any ring deactivating group present. For example, the aromatic hydroxylation of diazepam is occurring in the activated ring at the fourth side. So these are the two rings. You may observe that in this ring chlorine is present, and in our previous slide we have discussed this chlorine is basically one of the ring deactivating group. So at this ring there will be the lesser chances of the aromatic oxidation. But on the other hand, if we observe this ring, this aromatic ring does not contain any of the chlorine or other ring deactivating functional groups. So the aromatic hydroxylation can take place at this ring at this fourth number position. The next is the oxidation at the carbon-heteroatom system. So these are the type of phase 1 oxidation reaction that involve the oxidation at those places in the chemical structure of the drug where carbon is being bonded with the heteroatom. It can be carbon nitrogen, carbon sulfur, or carbon oxygen. So basically these oxidation reaction can further be divided into different type of other reaction, like the site involving carbon nitrogen can cause the N-dealkylation, oxidative deamination, formation of N-oxide or N-hydroxylation. Now let's discuss them one by one. N-dealkylation. N-dealkylation is occurring in those substrate or in those drug molecule in which the alkyl group is basically directly attached or bonded with the nitrogen atom. For example, methamphetamine, that is one of the antidepressant drug, where it get metabolized and converted into the amphetamine drug molecule. Amphetamine is basically metabolite of methamphetamine via N-dealkylation. Methamphetamine, the name itself indicates meth is basically showing the presence of methyl group in its chemical structure at a particular site. So after phase 1 oxidation, this methyl is get removed. So this is the dealkylation reaction. Amphetamine will then further get metabolized to the phenyl acetone and ammonia via oxidative deamination. So this is another type of oxidation reaction that occur after this dealkylation in the methamphetamine. The other example is the conversion of meth barbital to the barbital. So the meth barbital have the methyl functional group that is attached at one of the sites and directly attached with the nitrogen in their chemical structure, and after metabolism phase 1 reaction it will undergo through the N-dealkylation, that is the basically demethylation, because removal of methyl take place, and now the drug will get converted into the barbital that lacks the methyl group. The dealkylation of secondary and tertiary amine will ultimately get converted them into the primary amine and secondary. So secondary amine, whenever undergo through the dealkylation, they get converted into the primary amine, and tertiary amine will converted into the secondary amine. The proposed mechanism for those are basically the N-dealkylation abstracting the alpha hydrogen atom. The typical N substituent removed by the oxidative dealkylation can be the methyl, propyl, and propyl isopropyl. Usually N dealkylation initially occur with the smaller alkyl group. Substituents that are more resistant to the dealkylation include the tertiary butyl, because they do not have any type of alpha hydrogen. And beside the cyclopropyl and methyl cyclopropyl are the cyclomethyl. So basically the dealkylation reaction can occur in the secondary tertiary amine to convert them into primary and secondary amine. Clear? So now the type of N substituent means the type of alkyl group that are directly bonded with the nitrogen in the chemical structure of drug molecule are mainly the smaller alkyl group, but that is the methyl group. But beside methyl they can be ethyl, N-propyl or isopropyl. There are certain bulky groups that are more resistant to the N-dealkylation are the cyclopropyl and the butyl group. S-oxidation reaction. S-oxidation is also one of the oxidation reaction that is occurring at the carbon sulfur heteroatom site in the chemical structure, or what we say the sulfide unit. There are the different example of the drug like anti-diabetic drug, antihistamine and antipsychotic drug that undergo through the S-oxidation phase 1 reaction to get converted into the sulfooxide metabolite. The barbiturate drug that is like thiopental undergo conversion into the pentobarbital via desulfation. This shows that whenever drug undergo through the S-oxidation reaction, so what is happening, basically the sulfur is being oxidized, the sulfur is being removed or get converted to sulfur. But beside this, the removal of sulfur can also take place, that is the desulfation, where the cleavage of carbon sulfur double bond can occur and it will converted into the carbon oxygen. The S-oxidation reaction is occurring in all those drugs that contain the sulfide unit. The example of drug that undergoing through the S-oxidation reaction are the methimazole, that is the antithyroid drug. Among the antihistaminic drug, that is the cimetidine, ranitidine, and chlorpromazine. Thiazolidine are basically the one of the important class of anti-diabetic drug. They contain the sulfur because thiazole is showing the presence of thiazole ring, and that contain the sulfur as a heteroatom. They will undergo through the S-oxidation reaction for their phase 1 metabolism. O-dealkylation. O-dealkylation is basically refers to the removal of heteroatom that is attached with the oxygen in their chemical structure. The example of drug that undergo through the O-dealkylation are the phenacetin, that is one of the analgesic drug, codeine, that is analgesic drug. So these are metabolized by O-dealkylation. Beside this, some drug undergo through the oxidative dehalogenation, that is the removal of the halogen molecule. And among them the halothane, general anesthetic, can sometime undergo through the metabolism by the oxidative dehalogenation, but halothane can undergo the other type of reaction as well. Phase 1 reduction reaction. Phase 1 reduction reaction is basically the reduction reaction, it is the gain of electron. The reduction reaction results in the generation of polar functional group like the amino hydroxyl functional group, which may undergo further through the phase 2 reaction. Clear? So this reaction occur at the several functional group like carbonyl, like hydroxyl functional group. The reduction reaction can take place in the different type of drugs that contain the aliphatic, aromatic, aldehyde or ketone functional group in their chemical structure. The example of drug like one of the analgesic, that is the methadone, sedative and hypnotic, that is the chloral hydrate, naltrexone, that is used for the management of alcohol poisoning. All these drugs are basically undergoing through the phase 1 reduction reaction for their metabolism. So this diagram basically explaining the reduction reaction. The reduction reaction takes place mainly in those drugs that contain the type of nitro, azo or N-oxide functional group in their chemical structure. For example, the nitrazepam, that is one of the benzodiazepine, it contains the nitro group in their chemical structure. Clear the proton cell, that is one of the antibacterial drug, it contains the azo linkage or azo bond in their chemical structure. This is the diagram for the azo linkage. Azo linkage is that linkage in which two nitrogen atoms are bonded or connected with one another by the help of double bond between them. And beside this, both of nitrogen may have the different alkyl radical. These are basically representing any type of aromatic ring or other heteroaromatic ring or alkyl side chain. So basically after the reduction, these nitrogen containing group like nitro, azo and oxide are get converted into the amine. The halogen atom present in certain drug can also undergo through the reduction reaction, like a halogen atom present in the halothane. I have already discussed in previous slide that halothane can undergo through oxidative dehalogenation or other type of reaction, that is basically the halothane and reduction. Phase 1 hydrolysis. Phase 1 hydrolysis is also one of the important type of the phase 1 oxidation reaction. In this basically hydrolysis of the drug take place. Hydrolysis of drug take place mainly in those drugs that contain the ester and amide drug. And ester and amide, we know that these are the important class of the product that are the carrier link product. So upon hydrolysis, these esters get converted into the carboxylic acid and then will undergo through the further process to convert it into alcohol. Mostly the hydrolysis reaction are get catalyzed by the different type of enzyme like esterases and amidases, depending on the type of ester or amide present in their chemical structure, but mainly it is catabolized by the esterases. The mostly esters are administered as product which on hydrolysis get converted into their active form because they have to produce their pharmacological activity. For example, the aspirin occur as an ester. Its pharmacological activity is an analgesic and antipyretic. Now, as I have discussed, that there are the two types of the hydrolysis, mainly ester and amide hydrolysis. Amide functional group or amide hydrolysis usually take place at the slower rate in comparison to ester. Why this is happening? Because of the availability of the esterase enzyme in the higher concentration, and because of their suitability for the hydrolysis reaction. The reaction in the secondary and tertiary amine occurs more readily, and in the primary amine it occurs rarely. For example, the procainamide, that is one of the antiarrhythmic drugs. Ester hydrolysis is catalyzed by the esterase enzyme that is present in the liver, kidney and other tissues where the metabolism can take place. And the non-specific esterases like acetylcholinesterase, hydrocholinesterase that is present in the plasma can also catalyze this type of ester hydrolysis. Amide hydrolysis is also catalyzed by non-specific esterases in the plasma, and beside this, the amidases is only present in the liver, like in the smooth endoplasmic reticulum of the liver. More specific enzyme systems are able to hydrolyze the sulfate glucuronate conjugate as well as the hydrated peroxide. Now let's have a quiz. So these are some basic questions from this lecture. For your self-evaluation, you have to attempt the answer of these questions in the comment section, and you may also ask the question relating to this lecture. Question number 1. Give the example of drug that undergo through the metabolism reaction of aromatic hydroxylation, reduction and hydrolysis. Question number 2. Briefly mention how different substituents can affect the aromatic hydroxylation.